can't join us for later, and yep, it's all yours.
Excellent.
Would it be helpful if I watch for hands raised and sort of let you know if that's the case as we go here?
That would be great. I won't be able to see anything. So.
Okay, yep, I'll keep an eye out.
Great. All right, so really my pleasure to be here. I think probably coming from a bit of a different part of the world than where most of you are working, but I think addressing some of the shared challenges. And so, yeah, as Alan said, the Blue Brain Project, it's actually a Swiss-based project, Swiss-funded project. And the goal is to digitally reconstruct the mouse brain and simulate it on supercomputers. And so it's been going for quite a while. In the midst of the Blue Brain Project, we also proposed and got funded the largest research grant ever for the Human Brain Project, which is wrapping up in about a year or so. And that was focused on organizing and integrating and simulating data about the human brain, where this one is really focused on the mouse brain. And so one of our core challenges in building any of these models is that neuroscience data is actually extremely heterogeneous. It's coming from all different levels of the brain, from the subcellular level to cellular level to tissue level to whole brain. And it's produced using so many different experimental techniques that it's extremely challenging to put all of those pieces together and understand how do they relate, how do they fit, and how do they tell us fundamentally, core insights about the underlying brain that's being measured. And of course, it's very often measured from many, many different individual brains because you can't measure all of these simultaneously from a single living brain. And so in the Blue Brain Project, we started with this first kind of proof of principle of how do we do this? How do we tackle organizing and integrating data about one single part of the brain, which is a neocortical microcircuit? And we had a whole bunch of data from the rat, from rat somatosensory cortex. And this was systematically measured over about 10 years and 20,000 experiments to characterize individual neurons, their three-dimensional structure, the way that they communicate between each other, all within this tiny piece of brain that's about 0.5 millimeter wide and 1.5 millimeters tall. And so it was really kind of the first, get the parts list of this little brain circuit, get all the information about how the genes are expressed, all the different types of cells, all the different synaptic properties, and organize that data. And then use a data-driven approach to kind of reconstruct the biologically dense circuit. We know that no matter how much data we had, it was always going to be very sparse compared to the biology in terms of describing the whole biological circuit. So we needed to use an algorithmic process to kind of reconstruct this three-dimensional circuit in the computer and then run simulations on the supercomputer. And the way that we did all of this was through a very iterative, integrative process. We had to organize the data, we had to build models, run simulations, validate, and then refine. And this is essentially an endless loop. As probably many of you are aware, you have to just keep iterating and learning as you go how to organize the data. How does that data inform the models? How do you improve the model building process? And then, of course, the validation informs your success and the next iteration.
Hey Sean,
yeah.
Are you advancing your slides? Because I'm not.
That's what I was afraid of. I am advancing the slides.
Oh, there! I just saw one. Yep. Is everybody else? I'm now on slide 7, but I missed.
Yeah, I think maybe I have to keep it in this view for that to work. So can you see this?
Yep. Sure. Yeah. Yep.
Okay. So these are the things I was talking about. Maybe just flip back quickly. So this is the very heterogeneous data from all different levels of the brain and lots of different experimental modalities. And our challenge is to be able to put all of that back together and get a better understanding of the brain circuitry. And then our first proof of concept, this is the neocortical microcircuit and all the parts that we measured experimentally in a lot of detail and lots of experiments. And then we had to establish this data-driven process. And that's that iterative cycle I was describing of organizing the data, building the models, running the simulations, validating, and then refining. And to do all of this, we built pretty extensive informatics infrastructure, from organizing the experimental data, to building the models, to running the simulations, to doing all of the analysis and visualization. That is particularly critical because you have to be able to relate, huge amounts of numerical data to neuroscientists to be able to kind of see things that are comparable to their experiments, and then do the validation in that way. So this required a significant investment in both custom software and hardware. And of course, our challenges from a data integration perspective are, of course, the data is coming from lots of different sources. Some of it's from literature, so we have natural language text mining, and then it's coming from different online databases, from labs that we collaborate with, and different brain initiatives. And that data is extremely heterogeneous. It's coming in lots of different forms. There's a lack of standardized ontologies, and major challenges to get everything kind of normalized to a point where you can actually meaningfully integrate that. And then, of course, challenges from the workflow perspective. How do we understand the context within which the data was generated? And of course, data also evolves as you're going through the course of a 10-year project, and it's captured using different workflows, and it's different equipment to do the measurements. And so a lot of challenges just to keep track of where that data is coming from and how it's being produced. And so, of course, we started the project with some relational databases, and rapidly learned that things change too quickly, that we keep busy redesigning our SQL database all the time. We get our data in Excel, and that also has all tremendous limitations. There's so many challenges, and we developed and threw out many different data solutions before kind of learning and thinking about, well, how could we build something that was flexible enough to integrate extremely heterogeneous data, something that would be able to scale, something that would be able to evolve as we needed to evolve our data types, and also our ontologies, because as we're going along, we're actually learning new categorizations of the building blocks of the brain. So the cell types need to evolve, the brain regions need to evolve. These things change as we get more data and more knowledge. And of course, in 2012, when we were planning out the Human Brain Project, we needed to also think of, well, how do we build a system that would work worldwide, that would be able to integrate petabytes of data, that would be distributed, and would essentially keep up with these challenges across many different sites. And certainly took a good bit of inspiration from Google's knowledge graph. And Google has this knowledge graph, of course, that also serves for data set search more recently, not back in 2012, but more recently they kind of released that. And what that highlights is, the kind of the best state of the art for finding data today is to go and do a Google dataset search. And what you find is, you get zipped files of lots of miscellaneous data in lots of different formats, and lots of different terms of use, and it's really challenging. And so even today, it remains a major challenge to actually usefully find reusable neuroscience data. And I think you're all familiar with structured and linked data standards and JSON-LD. Obviously, that was very useful for us, and the governance model of schema.org was very compelling as kind of a successful example of the community proposing and evolving standardized schemas for entities. But we needed something new, and we needed something, to really enable building a knowledge graph for neuroscience. And in the context of our project, we wanted our scientists to be able to answer different types of questions, like, what neuron morphologies or which data is available from primary somatosensory cortex in the mouse. What's the cell density? What are the data available to validate my model? Tell me everybody who contributed data and algorithms and model building pipelines to this simulation result, because that's important for academic credit. Things like that. And so we kind of modeled off of schema.org this NeuroShapes, neuroshapes.org. It's a community initiative for interoperable standard metadata models for neuroscience data. And that was based on the W3C Prov model, so capturing provenance, that was expressed in Shackle for validating the metadata and being able to know that, for which data do we actually have valid metadata, and then really in general adopting linked data principles to ease that federated access and discoverability of data across the federation. Another piece that we needed is that there was common minimal metadata that we had for every piece of brain data. We needed to, at least conceptually, be able to position that data in an atlas so that we would know, like, where in the brain is it, what species is it from, what type of data is it, who produced it, because that's actually a big part of just even understanding the value and quality of the data, where to access the data in a structured way, and then the license. Is it readily available? Is it something we'll have to negotiate access, or can't be used for commercial purposes? Different things like that. And so this minimal information for neuroscience data sets was an important development. And then we really needed a piece of software to support this in the context of those workflows I was describing. We needed to integrate that data. We needed, all of the existing data platforms for neuroscience are very, very domain specific. So you've got brain imaging data management, you've got molecular data management, all of these things, but they're all separate. They're all individual for their domains. There's no way to really integrate them, connect them, and at the same time support common data discovery and have access to the data in a really fair way, and findable, accessible, interoperable, and reusable. So this system, which I'll talk briefly about, is Blue-Brain Nexus. We've recently had this paper accepted, which describes it in a number of use cases. So it's in the Semantic Web Journal, should be coming out. It's there already, but it should be officially coming out any day now. And yeah, I don't think I'll go through that. This is kind of the stack. We've got Nexus Fusion, which is the interface layer, the web interface layer. We've got Nexus Forge, which is a Python layer for kind of interacting and building the knowledge graph and querying it. We've got some layers for building user interfaces, and then the back end is Nexus Delta. And Nexus Delta really manages that fair data store. It provides the data management layer, and it interfaces to, for example, supercomputing storage, cloud-based storage for any of the big binaries, as well as being able to reference, essentially any data source around the world through the web. The core model, it's built off of open source technology. So the primary store that we use is Cassandra, Elasticsearch for indexing, and BlazeGraph for the graph indexing. And it uses an event streaming model to basically ingest JSON LD, put that in the primary store, and then trigger a number of indexing processes to build and maintain and keep maintained the indexes, so that we then through a HTTP interface allow writing and updating those data, as well as all the queries necessary, and being able to build the custom Elasticsearch indexes that use the Blaze graph queries for the flexibility. So we basically can optimize any Blaze graph query by building a view that uses an Elasticsearch index to make that very, very fast. So Nexus Forge, as I mentioned, is essentially a Python SDK for building and using knowledge graphs. You use it to discover and reuse data models and schemas, and essentially really moving between the Python object world and your objects in the knowledge graph. It integrates with various backends, and of course, the primary one is Nexus, but it's designed to be independent of the backend. And it really enables that shackle validation and registration, and then searching and downloading data and query results into data frames for analysis. So we typically, when using Blue Brain Nexus, really start simple. You can bring your data in without a schema and then refine it later. You can describe your entities semantically if you need the semantics. Of course, you don't have to. And then we do support storing the actual binaries managed through Blue Brain Nexus with both local and remote data storage. But of course, you don't have to. You can always point to another data management system as long as it's got a valid URL for the location. And then it allows you to really establish project-specific security and enabling, who can have access to this project from which organization, to keep all of that secure, and then you can also build projects that essentially federate across projects within a Nexus instance and across Nexus instances, so that you can actually, as long as you have the permissions, you can build a federated view across these secure projects across different Nexus instances. So as I mentioned, the data quality and some of the governance is really established by defining a shackle schema, and shackle validation is built in to Nexus, and so that allows us to really apply both kind of core property constraints as well as semantic constraints. And then I think probably most of you are familiar with Provenance. Provenance, is really keeping track of, for a particular data artifact like a PDF, where was it? How was it generated? It was generated, maybe by a software agent from a particular data set, using a particular activity version, and maybe initiated by a particular individual. So provides a lot of very rich information about the provenance, where did that data come from, its lineage, the processes that generated it, and where the original data came from. And for us, and in the context of this project, it was really, really critical to know the origin of the data, to be able to track how the data is being used and transformed, to be able to assess the quality of the data based on the process that was used to generate the data, as well as who generated it, giving us some sense of also reproducibility of different workflows, so tracking which version of which code was used for transforming the data or for building a particular model. And then because it's a team science-oriented project, really being able to provide that attribution of who contributed to which project, which results, both in terms of data, algorithms, and just science. So we do think that Nexus, provides a pretty comprehensive implementation of the FAIR data principles, supporting persistent identifiers, high quality metadata, provenance, semantic search, federation, open API, secure, scalable, supporting multiple versions, supporting controlled vocabularies, open standards all the way, open source, and open to extensions. And then yeah, I mean, it's really supporting that evolution and migration of data as well as schemas and schema versions change. I mentioned this earlier, the schema.org and then neuroshapes.org is really the place where we've collaborated with a number of different organizations on standard schemas. I think this this piece is where we really learned something, I think, fairly deep about how to integrate very heterogeneous data. And in the context of this scientific challenge, we started to define our data types in the context of their provenance. And so we actually, this looks, really complicated, but we found that subtle details in the way that the data was generated. So for example, we have reconstructed neurons where somebody traces, a scientist and a scientific team traces the three-dimensional structure of a neuron. And the resulting data file is just called a neuron, right? It's a reconstructed neuron. And so you can go onto these online databases and you can find, hundreds of thousands of reconstructed neuron data files in standard formats. But there's a huge difference between a neuron that was reconstructed from a slice of brain in a glass dish, an in vitro slice reconstructed neuron, and a neuron that was labeled in the context of the whole brain and reconstructed using different imaging methods that never sliced the brain, right? Or sliced the brain in different ways and reassembled the data differently. So that became where we actually describe these provenance patterns for data types. And so this is just kind of a zoomed in view of that same thing, where we actually keep track of the individual protocols used to prepare the brain from the subject, which processes were used to actually label the individual neurons, what was the histology process to fix the tissue, how is the imaging acquired, all of this information that results in a reconstructed cell. And if any of these steps are significantly different, it changes the scientific meaning and intent or ability to reuse and combine with other data this neuron. So you really have to know this stuff because we're building electrical models of neurons from this data. And so it really, really matters. And this is just a contrast to a neuron reconstructed from a whole brain using a genetically modified animal and with whole brain imaging. And so it's a very, very different process. And then the other big piece that the knowledge graph serves is because this is a team needing to collaborate on one common goal, which is building these models, building a model of the brain, many, many different tools work on pieces of that problem. So there's curation of the literature, there's of course different data integration pipelines, there's a voxelized view of the brain, spatial indexes, command line interfaces, atlases, different encyclopedias essentially for different levels of the brain, like the ion channels or the neuron morphologies, and then different workflows for curating specific types of data, reviewing them, correcting things, asking questions, and then overall search. But all of those pipelines, all of those tools should be working off of a common and current representation of all of our data. And that's where the knowledge graph is that integration point for collaboration. And so we have to maybe play this manually. But so we build individual neuron morphologies, neuron models by integrating neuron morphology data, gene expression data, characterizations of individual ion channels, and then we use a machine learning algorithm to optimize, to recreate the behavior of individual neurons through that integration. And then we build model cortical microcircuits by, again, querying the knowledge graph for the composition of the different cell types within this brain volume, and then building a computational model It's three-dimensional reconstruction of that volume with all of the cell types, assigning the synaptic connectivity, and basically building up all of the dense reconstruction of that neural circuit. And then when we put all of that together and run simulations, we've assigned mathematical models that describe the voltage and current evolution based on all of the biological data. And that predicts essentially circuit level behavior that we then validate against data that is captured at the circuit level and other types of signals, including at the brain slice level, where many experiments are done where there's essentially a piece of brain is sliced. It's still a bit alive in some sense, and it's in addition you're recording activity, and we can compare from that between the biology and the experiment. But we can also compute from the simulation essentially what the brain waves that are measured in an experiment with a wire, putting an electrode into the brain tissue. We can compute the equivalent of that from the simulation and then compare the real experiments with the simulated through that computed brainwave in some sense. And so this is just a view where, rather than computing that brainwave for a single point, which is what you would see with a single wire, we can compute it for an entire volume in the brain circuit and then compare that to the biology. So it gives us a very valuable tool to bridge between cells and synapses in the circuit and things that you can measure in the lab or even in the clinic. This is just one view of the knowledge graph. We've experimented with different ways of being able to visualize projects and relations between different projects. In general, we don't really find these graph views to be useful at the user level, but they look kind of cool. Find it much more useful to actually build applications like Brain Atlas tools that actually use the content of the knowledge graph to then feed this web interface and be able to visualize, for example, all the different cell types, their densities throughout the brain and throughout a brain area of interest, or to be able to do queries and look at the neuron morphologies three-dimensionally and across the extent of the brain, and be able to perform analyses and questions about which brain regions do they intersect, do they project to, and so on. And then also in this context, being able to integrate those neuron morphologies from many different data sources from around the world. And then being able to feed brain building for larger brain circuits, like the whole brain, where we can populate the neuron morphologies and their models throughout different brain regions and build larger and larger circuits. And those are used then to feed simulations, large-scale simulations of the mouse brain, where we can simulate activity at much larger scales, and then again compare and analyze that in relationship to some of the large-scale imaging that's being performed in biological experiments. So Nexus, happy to answer questions. The Nexus is an open source under Apache 2. It's being used across quite a number of projects, and so it's quite generic. It's at the core of the eBrains, which is coming out of the Human Brain Project, the eBrains knowledge graph, which serves a large-scale neuroscience research infrastructure. I'm using it here in Toronto at the Krembil Center for Neuroinformatics at the heart of a mental health learning system. So we're actually able to adopt the HL7 FHIR data model natively within FHIR and then within Nexus, and then be able to use that to generate decision support tools for clinicians, as well as kind of creating a clinical knowledge graph and a research knowledge graph that can support different types of analyses. And then some collaborators, for example, the Switch infrastructure in Switzerland, it's a national academic infrastructure. They're using it to support the social sciences community in integrating and sharing their data across the country. And with that, maybe I take a pause and answer some questions. Thank you for your attention.
Yeah, really, really interesting stuff. And thanks for walking us through it, Sean. I'm curious about the politics of something like this and getting started. Given that you started with relational databases and Excel, for example, you know, how did people respond to what the proposal to use a knowledge graph, and how did you get critical mass to actually accomplish what you have?
Yeah, it's a really good question. I'm just trying to find my Teams. I guess this is the Teams window, but I only see white. So anyway, so yeah, I'll answer that question. I think it's a really important one. It's not easy. It's part of the challenge was that many different workflows had been built before we had the knowledge graph. And so there's a lot of resistance to re-engineering those. And the other side of it is that we had to spend quite a lot of time getting the scientists and engineers to see the value, right? Because especially if they're in general very comfortable working in their silo, except for a few key interface or integration points, then they're like, oh, what's the big deal? I can keep doing my stuff with my files in my project, and I'll just deal with it when I have to connect. But we've started to, we've really seen that change. One is because of top-down messaging that was like, look, this is going to save us a lot of frustration about where is the file for that, and when this team needs to access something, they can't find it. It's going to address that. But also exercising more and more frequently that integration cycle, it just becomes obvious to the teams that this is the only way to go. And so actually some of our strongest advocates for the knowledge graph became the lead scientists who are exercising those integration workflows. And so that became then, iteratively a very strong mandate. And it doesn't mean we have everything all fully integrated, but all of the core ones now really are. And so the circuit building, the circuit simulations, this sort of stuff is really working off of the knowledge graph and feeding back into the knowledge graph. And then we're working on some of the additional workflows.
Makes sense. And the other question I had was really regarding interoperability and reuse.
Yep.
And there's a dream of interoperability, and then there's the reality. There's all this, you mentioned all the different heterogeneous kinds of data that you're trying to work with. Are you successful in using a lot of other people's data for your purposes, or the scientists that you're working with? Are they seeing that kind of reuse capability?
Yeah, it's a very individual thing, and we have been able to, where we, for example, that neuron morphology use case, we are integrating and reusing the neuron morphology data from a number of labs. What we run into is that some data, even though they're very good at one level, for example, their completeness of the reconstruction across the brain, they maybe miss a key dimension, like they don't keep track of the diameter variations of the neuron, which actually turns out to be really important for the electrical modeling. And so we have figured that, well, for some of those gaps in the data where maybe some specific aspect wasn't measured, we can actually run some algorithms to fill in that information with reasonable approximations based on rules that we've discovered. But it's you know, at the scientific level, it takes very, very careful consideration of when is that data actually usable in the same way as another, and is it directly comparable? There's really never a case that you can just kind of plug in different data sources and immediately feel confident that that makes any sense to do, right? It takes deep kind of analysis of where did that data come from, how was it produced, what was measured with which quality, and then we do, you know, but we do have examples where that has been established. Also with the Allen Institute in Seattle, that, you know, they based a lot of their protocols, their experimental protocols for their large scale data production on protocols that we had pioneered. So there was enough compatibility there to be comparable. So it does happen.
Are others like the Allen Institute really plugged into how you've done this, and are they taking advantage of what they've observed that could be useful to them?
So I think that Allen certainly is, because they've also been, you know, kind of creating their own modeling pipelines, very much, you know, built off of and inspired by what we've done. And I think, as you know, there are other smaller groups that are building models that do reuse some of the core, but I think it takes time to really appreciate all of the different levels that you have to think about. And so, you know, it's happening, but I don't think it's a widespread, you know, it's certainly not a universal widespread thing. And we've certainly, you know, we've learned it's really, really hard to get independent labs that don't have the same needs, for example, to build a model from their data. It's very hard for them to find the motivation to actually produce the data at the quality level needed.
We had a question in the chat and then a hand up. I'm gonna read this, but it's a mouthful here. I'm wondering about the connection between so much heterogeneous data and the theoretical paradigms to support or refute hypotheses from the data. For instance, Jeff Hawkins' Thousand Brains model makes assertions about the homogeneity of human neocortex and proposes a general reference frame model for all forms of sensory and cognitive processing. I'm wondering how you might approach testing paradigms like this against all this data. Is there a model for paradigms?
Yeah, it's a really great question, and something that we've thought a lot about. So just with that specific example, one of the principles that we learned by building the models was that the circuits in the brain are not homogeneous. And where they sort of appear at one level to be very similar, because the neuron types in general seem to be quite similar. And then they behave similarly across different brain areas, and there's similar synapses and these sorts of things. But what we learned is that the wiring diagram of a local circuit depends critically on the composition of the cell types. And that composition, in terms of the relative percentage of different types of neurons, does change significantly as you move across the thalamocortical circuit. And so that means that the circuitry itself is changing significantly. And so it can be an illusion sometimes that it's like, you know, or it really depends, like, at what scale are you saying it's similar. So just to go a little bit further into an answer, you know, we do have the tools then to evaluate what does the reconstructed circuit actually look like from a wiring diagram, and is it similar, is it the same, what, based on which cell types or which criteria does it differ, and that gives us a framework for thinking about how circuits change in different areas, and testing hypotheses related to specific changes and how that affects the ability to process stimuli, for example, or inputs from other areas.
Very good. And then Michael had his hand up.
Yes, thank you. Very interesting talk. I was wondering, I have kind of two questions, a science question and a technology question.
Okay.
Science question relates to the example you had of the mouse brain, where you had simulation going on and all the different connections were lighting up here and there.
Yep.
And you're comparing that with the actual measuring of what's lighting up or not in an actual mouse brain, right?
Yep.
So my question is, how can you, I mean, it's almost, my first question is, well, how do you know what the mouse is thinking? But that's maybe a silly question. But what do you learn from comparing I mean, suppose it does turn out to be exactly the same.
Right.
You know, that it sounds exciting, but what's the real impact of that?
Right. Yeah, it's an excellent question. And fundamentally, one of the big challenges anyway is that you're never going to recreate exactly the activity of one mouse in your simulation, right? Every individual mouse is different, is unique, and it's also behaving and experiencing in its own context, right? With its own lifetime experience and this sort of thing. So there's no way that exactly the same thing is going to be recreated. And we're not going to know from this level what a mouse is thinking, right? But what it does do is it gives us a tool to, and it really is in collaboration with experiment, right? You can't just take a model alone and be confident of it. You're never 100% sure that the model is exactly right? You never will know. So it gives you a tool, though, to test out hypotheses that are hard to test experimentally.
Right. So you get an indication of whether it might be the right model if it's close.
Yeah, and then you use it to dive into principles. Right, that are general principles that you can then ensure that you can validate experimentally and then use to kind of advance our general knowledge of the brain. But
the next question is related to that, but it's kind of the technology side. So you're able to do this simulation and compare against it, but you could presumably do all that without the knowledge graph. So you made a comment that said, we'll use the knowledge graph for some things, but we're not going to put piles and piles and piles of data in there. We're going to point to the data. So my question then becomes, which I think is the correct thing to do. So my question is, the simulation piece, I'm guessing that's running outside the knowledge graph on the data that the knowledge graph points to. So what's the role of the knowledge graph in that simulation example? Maybe it plays a minor role.
Well, it actually is key to, so when we run a simulation, right, in order to configure the simulation, you need to read in lots of information, right? You need to read in lots of parameters, you need to read in cell densities, you need lots of information from the knowledge graph to build the model. And then once the model is built, we feed it off to the pipeline that takes it and runs the simulations. But to build it, like, that's the key thing. We need to be able to rapidly parameterize and build models of the brain and brain circuits.
Interesting.
With evolving data.
That's pretty cool. So just to summarize, you want to build models, but the models require lots of input data. The model can do all sorts of things, but 20 different models might still use the same input data. And the role of the knowledge graph is to have a central place to have that kind of data that anybody can use with any model they want.
That's right. That's right. And then to keep track, of, well, which simulations produced which results.
Oh, right. So all that is fed back. So you create the model all outside of the knowledge graph using knowledge graph information, but you point to the model in the knowledge graph and you point to the results of the model.
Yep.
Okay, thank you. That's pretty cool. Thank you.
We had one question in the chat, and then I'll get to you, Chris, next. How do you address reproducibility and sharing with respect to the meta-versioning concerns of all orchestrated digital artifacts that contribute to some conclusion or outcome, including source code, data, metadata, configuration, deployment?
Yeah. So it's been core in our thinking that basically, in principle, we should be able to fully reproduce any simulation artifact, right? Because we would know everything about everything that went into parameterizing the model, building the model, running the simulations, and everything. I have to be very honest. It's not there, right? We have the tools to do it at one level, but in terms of implementing it at all levels of all the pipelines, that's work in progress. On the other hand, there are specific pipelines and specific pieces of that where it's exact, right? Where we know exactly, for example, when we do our quality analysis of the morphologies and the repair of the neuron morphologies, and then we have an algorithm to actually synthesize new neuron morphologies from that data. All of that is exact. We know exactly the source of the distributions of the features that are used to parameterize the models that then synthesize the neurons. So we can keep track of all of that. It just takes where can you do it feasibly for which pieces, and then over time just keep getting better at capturing more and more of the details.
Cool. All right, Chris.
So this is something of a technical question.
Sure.
I'm wondering if you can say anything about the interface that you have to the knowledge graph. I think you mentioned a Nexus API. Is this kind of at the Sparkle level, GraphQL level, or does it have more content embodied in it?
Right.
More topic content.
Right. So we do have basically we do provide for each project a Sparkle endpoint and an Elasticsearch endpoint. So if you want to access it at that level, you can. And then we also provide basically a schema level, so you can request data structured according to a specific schema. Or data that is in that adheres to a specific schema, so you can basically directly request that through the REST API, and it is kind of a data-oriented API. We do like the GraphQL model, but we haven't yet gotten there. Like ideally, what we would like to do is actually provide a GraphQL endpoint to each project, and then be able to, according to the data types that are within that project, essentially have a GraphQL API
Right.
for each of those. We're not there yet. I would love to have that, but in the meantime, we basically get submit and retrieve JSON LD or triples or whichever form you want of the RDF from a direct REST call, essentially.
Okay, thank you. I mean, I guess it's a significant problem because the amount of complexity that you're querying on must be quite great, and doing that at a low level like Sparkle must be difficult. I guess I can see where your use of GraphQL might come in, but defining the schema would be quite a job, I would think.
Well, so we have schemas, right? We do have for all of our core data types, we have shackle schemas, and we can in principle use the shackle to generate a GraphQL schema, and then to essentially implement an API. I'm speaking a little bit too far ahead because that's where we want to go. But because we do already know the structure of the data well, that is possible. But currently, we allow both data to not have a schema, as well as then to be well constrained by a shackle schema, and to query it in either form. And then the Nexus Forge provides a way to also execute queries while hiding a lot of the Sparkle complexity, and then retrieving the data into a data frame.
Oh, thanks.
Yeah.
So what about the outlook for the project, Sean? Where are you hoping to go in the next few years in general?
Right. So right now the team is completely focused on our goal by the end of 2024 to have a full mouse simulation, full mouse brain simulation. So that's the goal, and all hands are on deck to basically ensure that everything to achieve that goal is in the knowledge graph, and is all the pipelines are updated and reproducible, and driving that single purpose.
Makes sense. And so how can the semantics community help you all achieve your goals? And, you know, is there a collaboration that comes to mind that more people in other areas, other disciplines might be able to contribute something? Because it seems like a very useful project.
It's a great question. It has been a very, so we have collaborated, for example, with you know, on developing what's called the Neuron Phenotypes Ontology, and this was a challenge to say, and it, you know, rather than just defining an ontology, it needed to be essentially a data-driven or very data-oriented ontology, because our neuron types are defined based on measured phenotypic properties. And so that was a really interesting challenge of how do we, like, there is the, you know, biomedical community cell types ontology, but that's much more on presuming that we know already the defining and differentiating characteristics. And here we needed them to be evolving. Essentially, what we came up with was a bucket of phenotype data functionally defining neuron types. So where, you know, this particular species, a particular brain region, a particular morphology type, a particular electrical type, a particular gene expression type, those become the defining phenotypic properties to help us, you know, say. So that was a very interesting collaboration, because it was kind of thinking quite differently about how do you build an ontology and for what purpose, and how do you interface that with kind of data-driven definitions of cell types. So I think opportunities like that, where we're just kind of shifting the game to more maybe data-driven and provenance tracking that data for the definition of the ontological entities.
Michael threw his hand up there real quick too. Go ahead, Michael.
Yeah, I'm intrigued by how difficult it has been to define the endpoint of what exactly constitutes a full mouse. I would imagine that's a bit of a gray boundary.
Yes, and basically, I mean, as with the microcircuit, we kind of had to be pragmatic and just say, here's our definition of a mouse, you know, of a microcircuit. So we had to come up with that definition. And in the mouse brain, obviously, you know, some people legitimately would say, well, a mouse brain without a spinal cord is not a mouse brain, because the spinal cord is so important. We basically are saying, look, it's really this central nervous system. It comprises, you know, these core areas, the cerebellum, the cortex, the thalamus. You know, we're not going to have all of the detail of some of the brain stem area, just for real lack of the detailed data that's necessary. So we're, you know, it's kind of a you know, gross level macroscopic view on what comprises a mouse brain, but it's
sure,
you know, pragmatic.
Okay, thank you. Yeah, I figured so. Thank you.
Another hand up from Greg.
Yeah, hi, thanks, Sean. I asked that previous question, but I'm looking at your slide there, your last bullet about building a community and joining us. I'm really fascinated in this work and have a lot of ideas about sort of more abstract pattern-based approaches to the interoperability issues of sort of data wrangling here.
Oh, cool.
And the modeling issues as well. And wondering what getting involved, joining us might look like in terms of what opportunities are out there to engage with the group.
Yeah, that's great. I think at one level where that text comes from is if anybody finds Nexus useful as a software stack, right? We're very happy to have contributors to that open source community, and then it certainly would be interesting if you have some thoughts on the interoperability and some of the integration challenges there. It would be great just to have a follow-up conversation and see what opportunities there are. It's obviously we're deep in it. We have 2 years left, so we're kind of deeply involved in I think some very specific ideas on the pipelines. But we're very happy to connect and discuss some of the challenges and details there and see if there's opportunities to collaborate.
Yeah, definitely. I can try to connect through LinkedIn. And to follow on Michael's question there about the simulation of the mouse brain, then. So can you just paint a quick picture of what that will look like, the experience of that simulation?
Right. So I think, as always, you have to define, well, what's the context that you're actually looking at? You can't simulate a mouse brain under all conditions, and so what we typically do, and what we've been doing so far, is identifying key experiments that we would want to recreate. Key experiments that would be kind of both a validation and used as a refinement target. So we would identify, well, what additional data is necessary to refine the model to recreate some core phenomena. And then we'd also look at, well, what are the experiments that have been published that maybe we don't know the mechanism, but we could actually run that and check in the model. And then what simulations could we run that would go beyond what's possible experimentally and make novel predictions that for the moment, maybe are not experimentally addressable or would be interesting targets for new grants, for example, to go in and run those experiments where possible.
Great, thank you.
Yeah, just to add to that, I mean, I think one key point to remember is we're really not claiming that this is the perfect model of every detail of the brain. This is basically our best first draft of integrating everything that we know today in order to build the best model today of the mouse brain, but that it should serve as a foundation for future one, guiding future experiments, but also providing an anchor and a foundation for integrating new data.
Yep. All right. Well, we are 1 minute over here, so we'll go ahead and call it. Thank you for kind of the update on the project and passing this along. I'll put the recording out for those that want it later. And is that the best way to connect with you, Dr. Hill, just on LinkedIn? Or?
Yeah, that's perfect.
Okay.
Great.
Okay. Very good. Well, thank you very much.
Thank you.
Yeah, this is super. We were happy to get the chance to finally hear from you and hear more about the project, because it really was compelling. So congrats on the successes, and look forward to seeing more from you.
Wonderful. Thanks so much for having me. Great to meet everybody. Take care.
Thanks.
Bye now.
Bye-bye.